practice problems. Let's start with terminology. The word aerobic means without oxygen. True or false? Hopefully you answered false to this question. Aerobic means with oxygen. Anaerobic means without oxygen. Here's a simple math question. Let's say you start with a glycogen molecule, and that glycogen molecule is going to undergo anaerobic glycolysis. How many ATP are you going to produce at the end of anaerobic glycolysis? Now the correct answer here is that you're going to produce 3 ATP. Now did you put 2 ATP? If you did, you were thinking about starting with a glucose molecule. Now remember, we have to put 2 ATP in at the start of glycolysis if we start with a glucose molecule. If we start with a glycogen molecule, we only have to put 1 ATP in. That's important because we're going to generate 4 ATP in the process of anaerobic glycolysis. When you start with that glucose molecule, you had to put 2 in, so your net is only 2 ATP. But if you start with glycogen, you get a slightly higher yield because you only had to put that 1 ATP in. You're going to end up with a net of 3 ATP at the end. One of our many G words is gluconeogenesis. So 1, you have to remember what this means, but 2, where does it primarily take place? Now the key to this question was where does it primarily take place? The correct answer should have been the liver. That's primarily where we're taking non-glucose precursors or non-carbohydrate precursors and we're converting them into a glucose molecule. Often that's happening in the liver. However, it can also happen in the muscle. It's just not the primary site of where that's occurring. However, it does occur in both locations. Here's a question about our carrier molecules. NAD is in the reduced state, and NADH is in the oxidized state. True or false? Hopefully you answered false. These are flip-flopped. So NAD is going to be in our oxidized state, and NADH is going to be in our reduced state. So NADH received the 2 hydrogen and electron molecules, so it's the reduced molecule. Now, as we've mentioned before, we can utilize protein as a fuel source. So we can take those proteins, specifically those amino acids, and they can undergo gluconeogenesis in the liver to produce glucose. They can also be converted to acetyl-CoA molecules. But it's really tricky to calculate how much ATP are we able to produce from a protein. Partly because we have to monitor the nitrogen excretion within the body to be able to determine how much protein is being broken down, but that process alone requires us to put ATP into the system. So now we're trying to balance how much ATP is going in to excrete this much nitrogen, and honestly it ends up usually being a pretty negligible amount. That's why in exercise physiology, when we're trying to estimate energy needs based on different substrates, we often just kind of ignore the protein. And we know that they play a role. The key is they just play a small role. Lactate can be an effective fuel source for our muscle during exercise. What you can see here on the screen is how pyruvate is converted to lactate. And importantly, we have to put in an NADH molecule in order to convert it from pyruvate to lactate. So we lose that high energy carrier molecule in the process that was produced by anaerobic glycolysis. The enzyme that's facilitating this transaction is lactate dehydrogenase, and it's the enzyme that can allow lactate to go in either direction, lactate to pyruvate or pyruvate to lactate. Notice the arrows go both directions. Within our muscle, there are 3 ways that we can utilize this lactate, and this is something that we've more recently learned quite a bit about. Option number 1 is that lactate can be shuttled into the mitochondria because remember it was produced in the cytosol where anaerobic glycolysis is taking place, and it can be directly oxidized for ATP. This is the number 1 most efficient way to utilize that lactate. And it's actually how we utilize most of the lactate that's produced in the muscle. We didn't know that 10 years ago. So this is rather new that this is pretty well supported by the research. Option 2 is the lactate can be shuttled out of an individual cell to another cell and then oxidized. This takes time. It's far less efficient from an energy standpoint. And option 3, the one we've known about for quite some time, is the Cori cycle. So we can actually take that lactate, convert it back to pyruvate in the liver. So we're shuttling the lactate out of the muscle, transporting it in the blood, sending it to the liver to be reconverted. This is not an effective mechanism. It costs quite a bit of ATP to go through this process. So luckily, we've now discovered that option number 1 is how most of the lactate is actually utilized in the muscle in an energy efficient manner to produce ATP. But we're going to go into a little bit more detail and look at how the Cori cycle, which is a form of gluconeogenesis, actually takes place. This slide gives a nice example or graphic of what the Cori cycle looks like. So we go through anaerobic glycolysis and we end up generating 2 lactate molecules. And that's either from starting with a glucose or a glycogen. We'll have those 2 lactates. The 2 lactate molecules are shuttled out of the muscle into the bloodstream and taken to the liver. In the liver, they undergo gluconeogenesis. So those 2 lactate molecules are converted to 2 of our pyruvate molecules, so going backwards, which will then be converted to glucose molecules. But here's the caveat, and what should be catching your eye in yellow. It requires 6 ATP to go through this process. Remember, we only generated 2 ATP when we went through anaerobic glycolysis. So the process of recycling these lactate molecules actually costs us energy in the end. This is a great summary slide that shows the 2 primary substrates, fatty acids and glucose, and it shows how they enter the muscle and then how are they metabolized in order to support ATP production, as well as where are they stored whenever they're not being used for metabolism. So take a few moments and pause your video and make sure you can follow the different arrows for each substrate and understand where it's going to supply energy and where it's going whenever it's in a storage form. An important segue into chapter 4 is to talk about the relation between duration and intensity of exercise and how we're generating ATP, either through aerobic or anaerobic means. When exercise intensity is very short and of very high intensity, we are primarily generating that ATP from anaerobic mechanisms. Whereas when we're doing activity that is very long in duration, obviously the intensity is going to have to be lower. We're primarily generating the ATP in that case through aerobic metabolism. In the figure on the right, you see some different examples of time course and what's the relative contribution of aerobic versus anaerobic metabolism, as well as on the far right of that figure, you see some examples of some actual athletic pursuits that would be happening or matching those different time intensities.